Again, welcome to the start of Chapter 2. This is dealing with a topic known as pharmacodynamics. A little bit of insight into how drugs actually do their thing once they get to where they're supposed to be going. Remember that as a healthcare provider, the best thing you can do above and beyond being really good in your particular niche is to have a broad understanding of what is going on in other niches, is to have some experience, whether it's academic or clinical, and you're going to gain experience clinically, but that's actually the long and sometimes difficult road compared to, say, academics. And that's the source of this quote right here. The art of healing comes from nature, not from the physician or, shall we say, the healthcare provider. Therefore, the healthcare provider must start from nature with an open mind. So I realize that many of you have not had biochemistry, you've had some physiology, and that's a good thing. What we're going to try to do is bring some of that to you, not so much for the drugs we can teach you about, but for the drugs that we couldn't imagine teaching you about, or some of the conditions that you might encounter that we can't predict. So hang with me today, and I will do my best to help you out as best I can. And as always, you can reach out to me with your questions. Let me know if you want to talk and do a face-to-face. That's a good way of interacting. It increases the ability to nuance answers and things sometimes. Chapter 2 covers one of the major pillars of pharmacology. If we think about clinical effect, if we think about pharmacokinetics, which we will talk about in Chapter 3, we also have what's called pharmacodynamics. And this is the study of or the understanding of what a drug does inside the body in terms of effect. What proteins or what molecules does it interact with, and what effects can we predict based on what we know. So just a little bit about the rationale behind having to learn about this topic. And again, this is a very, very cursory introduction to this stuff to give you a handle on what problems or how to overcome problems that occur in the clinic. You may not be directly responsible for solving those problems, but if you have some insight, you can then have a rational discussion. So pharmacodynamics is the study of the biochemical, physiologic, and molecular effects on the body, the downstream effects, the side effects, if you will, and potential chemical interactions. We sometimes call this metabolism. We sometimes call that ADRs, adverse drug responses, and so forth. Pharmacodynamics is going to help you be a better collaborator with the physician or the medical supervisor with a given patient. So it links physiology, it links that physiology that you've had with what the drug is doing inside the body. It helps explain the relationship between when we give a dose and the type of response that we see. When we have an effect, that yet almost always means that the drug is binding to some sort of a target molecule inside the cell that we call a receptor. It's not always the case, but that's 99% of the case. So it helps understand the relationship between dose and response. More is not better. More is more. And just like a homeostatic window in physiology, there is a therapeutic window for drugs. There's a side where there's not enough drug to have an effect, and there's a side when there's a drug that's having other effects that could be toxic or lethal. And how to discern between when we're in that homeostatic window, that therapeutic window, and when we're not. Some of the things that can affect pharmacodynamics on a general level that can be points of consideration when you're interacting or considering plans for a patient. The aging process has a profound effect on pharmacodynamics because we express different genes, and our genes do sort of develop mutations over time as part of the age-related process. Other drugs, we call this polypharmacy, and it is rampant in modern society. So we need you guys to be the eyes and ears when it comes to potential drug interactions. Other disorders, things like genetic mutations, like in the case of things like cancer, or perhaps they have a rare gene mutation, like you've ever heard of BRCA1. That's an example of a gene mutation that leads to elevated risk of cancer. Thyroid disease, malnutrition, myasthenia gravis. These are all just a few examples. Some forms of metabolic syndrome that we call insulin-dependent diabetes mellitus. These are all potential causes, and there's just a small list. So the list is extensive, so the more you can understand, the better off everybody will be. So this chapter is meant to give you context, to give you the background that you need. So like without context, information is just a piece of information is just a dot. It floats around your brain with lots of other dots. It doesn't mean anything, or worse, it could lead us to the wrong thing. So knowledge is information in context. So we're going to be connecting dots. And this will actually make your life easier. If you learn the basics and the backgrounds, this will actually make learning about new drugs a lot easier than it would be just trying to memorize them out of context. So this is a way of making things easier. So in order for a drug to generally have an effect, you have to have what we know of as a receptor, or sometimes we refer to that as a target. Some molecule, some structure, some aspect of cell function that the drug interacts with and causes some change, hopefully beneficial change. And these things are literally everywhere. With rare exception, there are some drugs that don't affect cells directly. They're affecting the chemistry inside the stomach or the digestive tract or the urinary tract. Those are mostly the exception. Almost all of the other drugs are affecting some biomolecule, some enzyme or such inside a cell. That can make things very complicated, but it can also make things relatively simple if you understand the generality that what we're doing is affecting cellular function. To give you a sense of how extensive drug targets are throughout the cell, there is actually a database online for looking at what we call potential drug targets. Targets that we know exist, or some that are potential, or that could be useful if we could ever find a drug that would affect that target. And so there are at least 1,200. Now this slide's a few years old, so it's probably higher than that now. But if you look at the potential ones, you can see that fully 80% are an enzyme of some sort. It means that they catalyze some type of reaction inside the cell. There are other what we call endogenous receptors. These are receptors for other endogenous molecules, antibodies, growth regulators. The sky's the limit, but you can see that fully 80% are some kind of an enzyme involved in some sort of metabolism. So bypassing a few steps here in terms of administration and so forth, we're going to just give you the short story about what can happen once a drug is inside the body. A lot of the times, the drug will move into the bloodstream and be transported in the bloodstream using a sort of protein carrier. Many drugs are relatively hydrophobic. Which means that they aren't very water soluble, so they need an albumin in the bloodstream, which has a hydrophobic interior to allow that drug to disperse throughout the body. Now, even if it isn't very water soluble, nevertheless it will cycle between what we call free drug in the bloodstream and drug that's associated with a protein carrier. So here's our carrier here, and we're going to have that interaction of free drug or bound, or we saw conjugated drug. Once that drug is free, it may diffuse out of the bloodstream and thus interact with either, say, a cell surface receptor up here, or perhaps it can diffuse through the cell membrane and interact with what we call a cytosolic or a cytoplasmic receptor. In both cases, those types of interactions frequently trigger a rapid type response. What I mean by rapid, it could happen in a few minutes or it could happen in a few hours. Rapid means it doesn't take days or weeks or that sort of a thing to happen. On the other hand, we may get drug that will penetrate all the way through the cytosol and actually go to the nucleus and bind a nuclear receptor. Again, this is another protein here. And if we bind a nuclear receptor, the odds are that nuclear receptor is going to interact with the DNA, triggering some transcriptional change inside the cell, changing which mRNAs are created, leading to a different suite of proteins, different abundance of different proteins outside the cell. That's a much slower process, minimum 24 to 48 hours, oftentimes days and weeks for that to manifest. But those are really the sort of the two major stories. We're either going to bind, something that leads to a rapid response, and that would be like an enzyme or a cell surface receptor, or perhaps we're going to affect something that changes the DNA. The real truth is, it's often both, but we're going to say either or. Either it's going to be some sort of a rapid or some type of a slow genetic transcriptional type of effect. So when it comes to drug targets, what do we mean by biological target? It means that we have some native protein, usually. I'm gonna amend that. Say protein or biomolecule in the body whose activity, if it's an enzyme or it's involved in a process, definitely the shape is changed or some other behavior of that molecule is changed. It's modified by a drug resulting in some kind of effect. You say specific, but in this case, it may be desirable or it may be an unwanted adverse effect. We're not discriminating between what we're trying to do and what actually happens. Biological targets are whatever that drug is going to interact with and affect in the cell. Now, I have this picture here. It's a little different from that potential target database. This is actually what we have that is actually what we call druggable. Means we have a compound and it can affect these targets in the cell. And these things include certainly proteins, a lot of things like G-coupled protein receptors, which we'll talk about in a few minutes. Almost half of the marketed drugs right now are affecting this G-protein coupled receptor, which is found on the cell surface. Over half of them. Why? G proteins are easy to target. They're on the cell surface. You don't have to get inside the cells. Certainly enzymes are out there, and the key with getting after enzymes is that drug has to be able to get into the cell. And that can be a challenge at times. You may have a wonderful drug that can be an awesome inhibitor of kinases or proteases and so forth, but because of its hydrophobic characteristics, you just can't get it inside the cell. Ion channels, when we talk about the neuroactive drugs, when we talk about the anesthetics and the analgesics, many of those go after, or the cardiac active drugs, many of those go after ion channels. And we have like chemically gated ion channels, and we have voltage gated ion channels. What we mean by gated is the stimulus that will trigger that ion channel to change shape, either open or close. Certainly, hormone receptors are a druggable target. Look no further than the testosterone supplements or postmenopausal hormone therapies. All of those are affecting nuclear hormone receptors and affecting genetic responses in the body. Structural proteins, we see these a lot, like for instance tubulin. That's a common drug target for certain forms of cancer. These things tend to slow down cell division and mitosis. Membrane transport proteins, and even we can target nucleic acids directly. There are a lot of different targets out there. The key is how do we manage the understanding of all those targets? And so what do? We generalize a little bit. So the general paradigm is that you have some sort of biologic target, a receptor, if you will, that receives a signal, and that triggers that receptor to change shape, change what we call conformation. That can then lead to subsequent steps, leading to a response/effect. That's the standard paradigm, whether you're talking about interacting at the cell surface or in the cytosol, or a nuclear receptor affecting the expression or turning down the expression of some DNA sequence. So some sort of input, some sort of integrating center, output, response. If you've seen physiology, it's the same story. This is what we call a scalable concept. If we look at this a little bit closer, here's our drug in question. It's going to bind a specific binding site in the receptor. So the receptor has a conformation, a shape, if you will, that allows it to interact in a generally non-covalent way. We're not going to assume covalent interactions, but we're going to interact in a non-covalent way that's going to affect the behavior of this molecule. Now, sometimes we have complications. For instance, maybe there are other molecules like this hexagon here that could also fit in that site and interfere with this drug. This could be an endogenous compound, or this could be some other drug. When we have two different molecules competing for the same binding site in a target or a receptor, we call this competitive binding. In this case, this guy is preventing the action of this. But the presence of this molecule is lessening the effectiveness of this molecule. And that's what that little T is there for. So if I draw that in there, anytime you see a line with a T like that, that means that it is inhibiting whatever process it's teeing into. Okay, so that means that this drug maybe can bind that site, but it doesn't cause the effect that the square one does. So it's a competitive inhibitor. On the other hand, we may have another site on that same target that's a different shape that allows another molecule to bind, again, non-covalently, generally speaking, that can then change the shape of this and make it less possible for this to bind. Or in some cases, more possible. That's why you have this sort of interaction going on here. This guy can affect how or why or if this molecule can bind. And we call that allosteric because it's not binding the original site. We see this sort of interaction. We have competitive inhibitors, if you hear that phrase. We hear non-competitive or allosteric inhibitors or enhancers binding a different site in that target. And then that will in turn influence whether or not we see agonism, which is the effect that we would predict, positive situations, or maybe we have some sort of negative effect, some shutdown or inhibition of response. Again, we're going to break this out a little bit. This is very, very generalized. It may be a little tricky to wrap your brain around these interactions just now. Start with the competitive stuff over here and the allosteric. We can get there, then we can move on to the later steps. But this is fundamentally pharmacodynamics. It's fundamentally right there. It's start with the simple stuff, and then we're going to add layers. One of the figures in your book shows the different types of receptors that we find on, say, the cell membrane surface, what the general response is, and then how long that typically takes to happen. For instance, we have certain types of ion channels that can also be receptors, and we call those ionotropic. So when the receptor is altered in its shape, it affects whether or not ions can get through. That affects the membrane voltage. Either we make it more polarized or we make it less polarized. And there are often voltage sensitive secondary proteins that when that voltage changes, it changes their behavior as well. These things happen super fast. A lot of the neuroactive drugs that we'll talk about affect ionotropic receptors. Cardiac, we have some ionotropic receptors as well, and muscle. This one's a little more complicated to explain, but a G-protein coupled receptor is a receptor that's associated with this second message called a G-protein. That's why we call it G-protein coupled. So there's a receptor molecule, And when it binds the drug or its ligand, it will release that G protein, and then that G protein is free to cause responses in other proteins. Like for instance, maybe it affects an ion channel, like before, causing a change in polarization and cellular effects. Or maybe that G protein affects an enzyme that's around, and that causes creation of other second messages to arise, whether it's calcium or we phosphorylate a protein, et cetera, leading to cellular effects. And this is a big one. Over half of the known marketed drugs right now are G protein coupled receptor type drugs. And we'll come back to this. Don't worry if you don't get it all in this slide, okay? You don't have to hang on my every single word for every slide. The third one here are what we call receptor enzymes, and many of these receptor enzymes are kinases. Like here, what they do is when the kinase is active, it will take an ATP and use that ATP to take one of those phosphates off the ATP and tack it onto the protein target. That's what phosphorylation means. We use ATP, you take that gamma ATP off and put it on this target, and now that protein's shape has changed and thus its activity has changed. These things are on the cell surface, a receptor enzyme, and it's a kinase. So we phosphorylate that target, and then we can have all kinds of cellular effects down that way. And then we have our nuclear receptors, and the most common drugs that affect nuclear receptors are going to be very, very lipophilic hormone type modulators. They behave like hormones. We ought to just call them hormones for all I care. In any case, the receptor is found way down inside the cell, in the nucleus. And when that receptor is bound by the drug or the molecule, it affects which genes are turned on and off. These two things are the same here. This one just happens to have a receptor inside the cell. This one happens to have a receptor on the same surface, on the cell surface. Again, these things take a long, long time. Estrogen receptor, they can take hours or days or even weeks, and so forth. We're going to see examples of these. You might want to be referring back to this from time to time if you're sort of losing track of the language, if you know what I mean. A good reference picture. So what I'm going to do now is show you some of these membrane receptors in action. So I'm going to drill down into these different categories so that you can see some examples of what we were just talking about to help you better understand those generalities. And we're going to start over here with the ionotropic receptors, the receptor channels. Again, here's our molecule. It may be a drug or it may actually be an endogenous molecule. Binds the channel and affects permeability to certain types of ions. Now I'm going to move through this because this is not the most easily understood figure. But what we're showing you here is how a drug can affect an ionotropic receptor. In this case, this is called the voltage-gated sodium channel. It actually has two gates, and they're both sensitive to changes in membrane voltage. You have an activation gate and you have an inactivation gate. So under resting circumstances, that activation gate is closed, and so sodium cannot diffuse down its concentration gradient. It's blocked from moving across the membrane. However, if we achieve a certain voltage in the membrane, a little bit of depolarization, and we hit a level called threshold, notice how these two subunits have now spread apart. They're further apart than over here. And so the activation gate has opened, and now sodium is free to move in and diffuse down its gradient, causing further depolarization as we have positive charges here. That further depolarization will eventually trigger an inactivation gate, which then closes over here on the right. So we're open right now, but then when we hit a certain voltage, it will close even though the activation gate is still open. So that's the inactivation gate closing, and now sodium is prevented from moving, or more sodium is prevented from moving into the cell. That's the normal behavior. If we add a blocking drug, and I don't want to get too much into the detail here, that blocking drug will actually come in and prevent sodium from coming in even though the activation gate is open. Even if the activation gate and inactivation gate is closed, still the same process. We have inhibited the ionotropic function of this channel. And I'm going to show you a few other examples over the next couple slides. Just another example of an ionotropic receptor. In this case, this is the acetylcholine receptor. And so you can see where the acetylcholine molecule binds, and it changes the diameter of this pore, allowing generally sodium to move through the plasma membrane. And here's where we would see those acetylcholine receptors. We would see them on the surface of a muscle. Occasionally, we'll see them on the surface of a neuron as well. So once that acetylcholine is released, those receptors open, let sodium in, and triggers an action potential in muscles. Very, very desirable target for neuromuscular relaxation, but also a target for certain types of pathology. And so we're going, by understanding that what's going on here with those acetylcholine receptors, we can now be more smart about therapeutic options. And just a few others. So we have some ligand types of channels like acetylcholine, the nicotinic receptor. We'll talk about that with sympathetic, parasympathetic. So it's gated for acetylcholine. It's also gated for nicotine. We have the GABA receptor, gamma aminobutyric receptor, very common in the central nervous system. Again, that's the amino acid it's sensitive to. Glycine's another one we're going to talk about a little bit later on this semester as well. Serotonin, we'll talk to neuroactive drugs. All of these things are going to be receptors that we will talk about. So getting familiar with the language of receptors, getting familiar with what these receptors are doing can and does make it easier for you to understand what these drugs are going to do, and importantly, what their potential side effects might be. Just a quick slide here. This is a book figure. What this is showing is called the patch clamp technique. So if we have what we believe to be a receptor of some kind, what you can do is you can isolate that membrane, and you can test that receptor by adding or affecting drug right on that area and looking for changes in voltage or so forth. It's called the patch clamp technique. The book talks about it just a little bit briefly. Next, we'll do some examples of G protein coupled receptors, a little bit of overview, and then just a couple of examples for you to see. G protein coupled receptors are very easily recognized because they have what's called a receptor domain. And a domain means a region of functionality or a subunit. And the domain for the receptor of a G protein coupled receptor crosses the membrane 7 times. And that's why they're very easy to spot. Here is the G protein over here. And what happens is when an agonist binds the receptor, it frees up that G protein to go do signaling downstream, usually quite quickly, within minutes or even seconds in some cases, but usually in the minute time frame. Lots of neuroactive drugs use G protein coupled receptors to modulate permeability across the membrane. So not as fast as the ionotropic receptors, but nevertheless a very important category. So we get a second message, that's the G protein, that leads to changes in protein behavior inside the cytosol, leading to some kind of a cellular response. So again, here's the receptor side of it. We have the ligand. In this case, this would be the molecule that normally binds the receptor, or it could be a drug if it mimics the ligand. Once it does that, it releases that G protein, which can then go out and signal downstream. So this is the unbound form right here. You see the G protein, and then here's the bound form, and now that G protein is out and on its way, causing a second message and responses. And lastly, for this lecture today, we have the nuclear receptors, which we have already talked about just a little bit, so I won't belabor it too much. Nuclear receptors oftentimes are residing in the nucleus before they're bound to the ligand or maybe perhaps a mimicking drug. There are, though, cytosolic versions of these. The receptors will be in the cytosol, and once they bind their ligand or drug, they'll move to the nucleus and then affect DNA expression. So we call these receptors, these nuclear receptors, DNA coupled. That's because they are fundamentally transcription factors. They alter the abundance of mRNAs as a response once they bind and interact with DNA. Maybe some mRNAs they're going to make more of, maybe some mRNAs they're going to make less of. So they affect the transcription and the creation of proteins. The DNA receptor, DNA coupled receptors, and the ligand generally must be pretty lipophilic. So we're talking about very, very hydrophobic substances, which there are a lot of pharmaceuticals that do that. But hormones like steroid hormones, things that have a lot of ability to penetrate cell membranes are usually in this category. So steroids, like I said, trying to think of anything else right offhand, but they would have to be something that can penetrate membranes if they're going to reach these receptors. It's just another picture here, just the last picture of the day, showing that sort of, again, that we have to have carrier proteins for a lot of our drugs, because for drugs to target inside the cell, they have to be able to get through the plasma membrane, which usually means they're hydrophobic. The vast majority of our pharmaceuticals that are orally active are somewhat hydrophobic, or they say lipophilic. That's another way of saying the same thing. They will bind a carrier protein. By the way, while it's here, while I'm thinking about it, this carrier protein, if you have drugs that compete for sites in that carrier protein, that's one of the common causes of drug-drug interaction. So if these sites are occupied by another drug, now that molecule that would normally reside inside a carrier protein for a period of time basically gets forced out into other compartments by virtue of its hydrophobicity, and now you start to see toxicities and side effects. So that concludes part 2A. We're going to switch on to part 2B in the next video. Thank you for your time, and I will talk to you soon.